Modification with CRISPR was inspired by bacterial defense systems. Bacteria defend themselves from viral attacks by stealing strips of the invading virus's DNA, which they then splice into their own DNA. These newly formed sequences are called clustered, regularly interspaced short palindromic repeats, or CRISPR. The bacteria then make RNA copies of these CRISPRs that help recognize virus DNA in the event of future invasions. Think of CRISPR RNA as a most wanted poster for viruses, with the bounty hunters being enzymes called CRISPR-associated proteins, or Cas. In 2012, scientists turned CRISPR from a bacterial shield into a gene editing tool. They discovered that they could replace the bacterial CRISPR RNA system with a modified guide RNA to act as new wanted posters. Once inside a cell, a bounty hunter enzyme called Cas9 scans the cell's genome until it finds a DNA match. It slices through the DNA, and the cell's enzymes then rush in to repair that damage. At this point, scientists can change or add DNA within the sequence. This is a huge deal and gives scientists a powerful new tool for gene editing. Best of all, it's really cheap. As research has progressed, improved techniques have made CRISPR-based gene editing easier and more accurate. CRISPR is already being used to edit pig DNA so their organs can be transplanted into humans. Plans are also underway to edit allergens out of peanuts, create mushrooms that won't brown, and breed genetically engineered mosquitoes that can't transmit malaria. But as Philip K. Dick might have wondered, the real question is what CRISPR can do to humans. On the less controversial side, it can change the genes in somatic cells, the ones that aren't passed on to your kids. This technique can be used to make immune cells that are better cancer fighters and can target illnesses like cystic fibrosis, sickle cell anemia, and Huntington's disease. But here's where things can get controversial. CRISPR can also change the genes in germline cells, or eggs and sperm. In other words, these are the changes that mom and dad can pass on to little Johnny and Sally. This method could wipe out genetic diseases like muscular dystrophy, and though it's pretty far-fetched now, these techniques could also hypothetically give people totally new abilities like infrared vision, improved athletic performance, and extra smarts. You know, like this guy, who's seen things you people wouldn't believe. It's the germline changes that worry scientists because we can't predict what every edit could do to a developing embryo. It also might worsen current inequalities through eugenics and discrimination, which is why many scientific organizations and countries oppose it. I can assure you there are a lot of ethical debates going on over genetic engineering, covering scenarios that were once just the stuff of science fiction. But rest easy, folks, CRISPR isn't ready for designer babies quite yet. It still has a long road of development, and this trip is just getting started. But we are heading down a bright path, one where we could wipe out certain genetic diseases completely. Needless to say, that is amazing. So what do you think, folks? Will CRISPR offer us a future not too dissimilar from Blade Runner, or will its potential be lost in time like tears?